Chapter 24 Fungi Figure 24.1 Many species of fungus produce the familiar mushroom, which is a reproductive structure. This coral fungus displays brightly colored fruiting bodies. This electron micrograph shows the spore-bearing structures of Aspergillus, a type of toxic fungus found mostly in soil and plants. Credit, mushroom, modification of work by Chris Wee. Credit, coral fungus, modification of work by Corey Zanker. Credit, Aspergillus, modification of work by Janice Haney-Carr, Robert Simmons, CDC. Scale bar data from Matt Russell. Introduction. The word fungus comes from the Latin word for mushrooms. Indeed, the familiar mushroom is a reproductive structure used by many types of fungi. However, there are also many fungus species that don't produce mushrooms at all. Being eukaryotes, a typical fungal cell contains a true nucleus and many membrane-bound organelles. The kingdom fungi includes an enormous variety of living organisms collectively referred to as eumycota, or true fungi. While scientists have identified about 100,000 species of fungi, this is only a fraction of the 1.5 million species of fungus likely present on Earth. Edible mushrooms, yeasts, black mold, and the producer of the antibiotic penicillin, Penicillium notatum, are all members of the kingdom fungi, which belongs to the domain Eukarya. Chapter outline 24.1 Characteristics of fungi 24.2 Classifications of fungi 24.3 Ecology of fungi 24.4 Fungal parasites and pathogens 24.5 Importance of fungi in human life 24.1 Characteristics of fungi. By the end of this section, you will be able to do the following. List the characteristics of fungi. Describe the composition of the mycelium. Describe the mode of nutrition of fungi. Explain sexual and asexual reproduction in fungi. Fungi, once considered plant-like organisms, are more closely related to animals than plants. Fungi are not capable of photosynthesis. They are heterotrophic because they use complex organic compounds as sources of energy and carbon. Fungi share a few other traits with animals. Their cell walls are composed of chitin, which is found in the exoskeletons of arthropods. Fungi produce a number of pigments, including melanin, also found in the hair and skin of animals. Like animals, fungi also store carbohydrates as glycogen. However, like bacteria, fungi absorb nutrients across the cell surface and act as decomposers, helping to recycle nutrients by breaking down organic materials to simple molecules. Some fungal organisms multiply only asexually, whereas others undergo both asexual reproduction and sexual reproduction with alternation of generations. Most fungi produce a large number of spores, which are haploid cells that can undergo mitosis to form multicellular, haploid individuals. Fungi often interact with other organisms, forming beneficial or mutualistic associations. For example, most terrestrial plants form symbiotic relationships with fungi. The roots of the plant connect with the underground parts of the fungus, which form mycorrhizae. Through mycorrhizae, the fungus and plant exchange nutrients and water, greatly aiding the survival of both species. Alternatively, lichens are an association between a fungus and its photosynthetic partner, usually an alga. Fungi also cause serious infections in plants and animals. For example, Dutch elm disease, which is caused by the fungus Ophiostoma ulmi, is a particularly devastating type of fungal infestation that destroys many native species of elm, Ulmus sp. by infecting the tree's vascular system. The elm bark beetle acts as a vector, transmitting the disease from tree to tree. Accidentally introduced in the 1900s, the fungus decimated elm trees across the continent. Many European and Asiatic elms are less susceptible to Dutch elm disease than American elms. In humans, fungal infections are generally considered challenging to treat. Unlike bacteria, fungi do not respond to traditional antibiotic therapy, since they are eukaryotes. Fungal infections may prove deadly for individuals with compromised immune systems. Fungi have many commercial applications. The food industry uses yeasts in baking, brewing, and cheese and winemaking. Many industrial compounds are byproducts of fungal fermentation. Fungi are the source of many commercial enzymes and antibiotics. Although humans have used yeasts and mushrooms since prehistoric times, until recently, the biology of fungi was poorly understood. In fact, up until the mid-20th century, many scientists classified fungi as plants. Fungi, like plants, are mostly sessile and seemingly rooted in place. They possess a stem-like structure similar to plants, as well as having a root-like fungal mycelium in the soil. In addition, their mode of nutrition was poorly understood. Progress in the field of fungal biology was the result of mycology, the scientific study of fungi. Based on fossil evidence, fungi appeared in the Precambrian era, about 450 million years ago. Molecular biology analysis of the fungal genome demonstrates that fungi are more closely related to animals than plants. Under some current systematic phylogenies, they continue to be a polyphyletic group of organisms that share characteristics, rather than sharing a single common ancestor. Career connection mycologists are biologists who study fungi. Historically, mycology was a branch of microbiology, and many mycologists start their careers with a degree in microbiology. To become a mycologist, a bachelor's degree in 610 Chapter 24, Fungi access for free at openstax.org. A biological science, preferably majoring in microbiology, and a master's degree in mycology are minimally necessary. Mycologists can specialize in taxonomy and fungal genomics, molecular and cellular biology, plant pathology, biotechnology, or biochemistry. Some medical microbiologists concentrate on the study of infectious diseases caused by fungi, called mycosis. Mycologists collaborate with zoologists and plant pathologists to identify and control difficult fungal infections, such as the devastating chestnut blight, the mysterious decline in frog populations in many areas of the world, or the deadly epidemic called white nose syndrome, which is decimating bats in the eastern United States. Government agencies hire mycologists as research scientists and technicians to monitor the health of crops, national parks, and national forests. Mycologists are also employed in the private sector by companies that develop chemical and biological control products or new agricultural products, and by companies that provide disease control services. Because of the key role played by fungi in the fermentation of alcohol and the preparation of many important foods, scientists with a good understanding of fungal physiology routinely work in the food technology industry. Enology, the science of winemaking, relies not only on the knowledge of grape varietals and soil composition, but also on a solid understanding of the characteristics of the wild yeasts that thrive in different winemaking regions. It is possible to purchase yeast strains isolated from specific grape-growing regions. The great French chemist and microbiologist, Louis Pasteur, made many of his essential discoveries working on the humble brewer's yeast, thus discovering the process of fermentation. Cell structure and function. Fungi are eukaryotes, and as such, have a complex cellular organization. As eukaryotes, fungal cells contain a membrane-bound nucleus. The DNA in the nucleus is wrapped around histone proteins, as is observed in other eukaryotic cells. A few types of fungi have accessory genomic structures comparable to bacterial plasmids, loops of DNA. However, the horizontal transfer of genetic information that occurs between one bacterium and another rarely occurs in fungi. Fungal cells also contain mitochondria and a complex system of internal membranes, including the endoplasmic reticulum and Golgi apparatus. Unlike plant cells, fungal cells do not have chloroplasts or chlorophyll. Many fungi display bright colors arising from other cellular pigments, ranging from red to green to black. The poisonous Amanita muscaria, fly agaric, is recognizable by its bright red cap with white patches, figure 24.2. Pigments in fungi are associated with the cell wall and play a protective role against ultraviolet radiation. Some fungal pigments are toxic to humans. Figure 24.2. Amanita. The poisonous Amanita muscaria is native to temperate and boreal regions of North America. Credit: Christine Mahul. Like plant cells, fungal cells have a thick cell wall. The rigid layers of fungal cell walls contain complex polysaccharides called chitin and glucans. Chitin, N-acetyl D-glucosamine, also found in the exoskeleton of arthropods such as insects, gives structural strength to the cell walls of fungi. The wall protects the cell from desiccation and some predators. Fungi have plasma membranes similar to those of other eukaryotes, except that the structure is stabilized by ergosterol, a steroid molecule that replaces the cholesterol found in animal cell membranes. Most members of the kingdom fungi are nonmotile. However, flagella are produced by the spores and gametes in the primitive phylum Chytridiomycota. Growth. The vegetative body of a fungus is a unicellular or multicellular thallus. Unicellular fungi are called yeasts. Multicellular fungi produce thread-like hyphae, singular hypha. Dimorphic fungi can change from the unicellular to multicellular state depending on environmental conditions. Saccharomyces cerevisiae, baker's yeast, and Candida species, the agents of thrush, a common 24.1, characteristics of fungi. 611 fungal infection, are examples of unicellular fungi, figure 24.3. Figure 24.3 Candida albicans. Candida albicans is a yeast cell and the agent of candidiasis and thrush. This organism has a similar morphology to coccus bacteria. However, yeast is a eukaryotic organism, note the nucleus. Credit. Modification of work by Dr. Godon Roberstad, CDC. Scale bar data from Matt Russell. Most fungi are multicellular organisms. They display two distinct morphological stages, the vegetative and reproductive. The vegetative stage consists of a tangle of hyphae, whereas the reproductive stage can be more conspicuous. The mass of hyphae is a mycelium, figure 24.4. It can grow on a surface, in soil or decaying material, in a liquid, or even on living tissue. Although individual hyphae must be observed under a microscope, the mycelium of a fungus can be very large, with some species truly being the fungus humungus. The giant Armillaria solitipes, honey mushroom, is considered the largest organism on Earth, spreading across more than 2,000 acres of underground soil in eastern Oregon. It is estimated to be at least 2,400 years old. Figure 24.4 A fungal mycelium. The mycelium of the fungus Neotestudina rosati can be pathogenic to humans. The fungus enters through a cut or scrape and develops a mycetoma, a chronic subcutaneous infection. Credit. CDC. Most fungal hyphae are divided into separate cells by end walls called septa, singular, septum, figure 24.5a, c. In most phyla of fungi, tiny holes in the septa allow for the rapid flow of nutrients and small molecules from cell to cell along the hypha. They are described as perforated septa. The hyphae in bread molds, which belong to the phylum Zygomycota, are not separated by septa. Instead, they are formed by large cells containing many nuclei, multinucleate, an arrangement described as conocytic hyphae, figure 24.5b. 612 Chapter 24, Fungi access for free at openstax.org. Figure 24.5 Fungal hyphae. Fungal hyphae may be a. septated or b. conocytic, coano equals, common, cytic equals, cell, with many nuclei present in a single hypha. A bright field light micrograph of C. Phyllophora richardsiae shows septa that divide the hyphae. Credit C. Modification of work by Dr. Lucille Georg, CDC. Scale bar data from Matt Russell. Fungi thrive in environments that are moist and slightly acidic, and can grow with or without light. They vary in their oxygen requirement. Most fungi are obligate aerobes, requiring oxygen to survive. Other species, such as members of the Chytridiomycota that reside in the rumen of cattle, are obligate anaerobes, in that they only use anaerobic respiration because oxygen will disrupt their metabolism or kill them. Yeasts are intermediate, being facultative anaerobes. This means that they grow best in the presence of oxygen using aerobic respiration, but can survive using anaerobic respiration when oxygen is not available. The alcohol produced from yeast fermentation is used in wine and beer production. Nutrition. Like animals, fungi are heterotrophs. They use complex organic compounds as a source of carbon, rather than fix carbon dioxide from the atmosphere as do some bacteria and most plants. In addition, fungi do not fix nitrogen from the atmosphere. Like animals, they must obtain it from their diet. However, unlike most animals, which ingest food and then digest it internally in specialized organs, fungi perform these steps in the reverse order. Digestion precedes ingestion. First, exoenzymes are transported out of the hyphae, where they process nutrients in the environment. Then, the smaller molecules produced by this external digestion are absorbed through the large surface area of the mycelium. As with animal cells, the polysaccharide of storage is glycogen, a branched polysaccharide, rather than amylopectin, a less densely branched polysaccharide, and amylose, a linear polysaccharide, as found in plants. Fungi are mostly saprobes. Saprophyte is an equivalent term, organisms that derive nutrients from decaying organic matter. They obtain their nutrients from dead or decomposing organic material derived mainly from plants. Fungal exoenzymes are able to break down insoluble compounds, such as the cellulose and lignin of dead wood, into readily absorbable glucose molecules. The carbon, nitrogen, and other elements are thus released into the environment. Because of their varied metabolic pathways, fungi fulfill an important ecological role and are being investigated as potential tools in bioremediation of chemically damaged ecosystems. For example, some species of fungi can be used to break down diesel oil and polycyclic aromatic hydrocarbons, PAHs. Other species take up heavy metals, such as cadmium and lead. Some fungi are parasitic, infecting either plants or animals. Smut and Dutch elm disease affect plants, whereas athlete's foot and candidiasis, thrush, are medically important. Fungal infections in humans. In environments poor in nitrogen, some fungi resort to predation of nematodes, small non-segmented roundworms. In fact, species of Arthrobotrys fungi have a number of mechanisms to trap nematodes. One mechanism involves constricting rings within the network of hyphae. The rings swell when they touch the nematode, gripping it in a tight hold. The fungus then penetrates the tissue of the worm by extending specialized hyphae called haustoria. Many parasitic fungi possess haustoria, as these structures penetrate the tissues of the host. Release 24.1 Characteristics of fungi: 613 digestive enzymes within the host's body, and absorb the digested nutrients. Reproduction: fungi reproduce sexually and/or asexually. Perfect fungi reproduce both sexually and asexually, while the so-called imperfect fungi reproduce only asexually, by mitosis. In both sexual and asexual reproduction, fungi produce spores that disperse from the parent organism by either floating on the wind or hitching a ride on an animal. Fungal spores are smaller and lighter than plant seeds. For example, the giant puffball mushroom bursts open and releases trillions of spores in a massive cloud of what looks like finely particulate dust. The huge number of spores released increases the likelihood of landing in an environment that will support growth. Figure 24.6. Figure 24.6 Puffball and spores. The giant puffball mushroom releases a cloud of spores when it reaches maturity. Credit A. Modification of work by Roger Griffith. Credit B. Modification of work by Pearson Scott Forsman, donated to the Wikimedia Foundation. Asexual reproduction: fungi reproduce asexually by fragmentation, budding, or producing spores. Fragments of hyphae can grow new colonies. Somatic cells in yeast form buds. During budding, an expanded type of cytokinesis, a bulge forms on the side of the cell, the nucleus divides mitotically, and the bud ultimately detaches itself from the mother cell. Figure 24.7. Figure 24.7 Budding in histoplasma. The dark cells in this bright field light micrograph are the pathogenic yeast Histoplasma capsulatum, seen against a backdrop of light blue tissue. Histoplasma primarily infects lungs but can spread to other tissues, causing histoplasmosis, a potentially fatal disease. Credit. Modification of work by Dr. Libero Aiello, CDC. Scale bar data from Matt Russell. The most common mode of asexual reproduction is through the formation of asexual spores, which are produced by a single individual thallus, through mitosis, and are genetically identical to the parent thallus. Figure 24.8. Spores allow fungi to expand their distribution and colonize new environments. They may be released from the parent thallus either outside or within a special reproductive sac called a sporangium. 614 Chapter 24, Fungi access for free at openstax.org. Figure 24.8 Generalized fungal life cycle. Fungi may have both asexual and sexual stages of reproduction. There are many types of asexual spores. Conidiospores are unicellular or multicellular spores that are released directly from the tip or side of the hypha. Other asexual spores originate in the fragmentation of a hypha to form single cells that are released as spores. Some of these have a thick wall surrounding the fragment. Yet others bud off the vegetative parent cell. In contrast to conidiospores, sporangiospores are produced directly from a sporangium. Figure 24.9. Figure 24.9 Sporangiospores. This bright field light micrograph shows the release of spores from a sporangium at the end of a hypha called a sporangiophore. The organism is a mucor sp. Fungus, a mold often found indoors. Credit. Modification of work by Dr. Lucille Georg, CDC. Scale bar data from Matt Russell, 24.1. Characteristics of fungi 615 Sexual reproduction. Sexual reproduction introduces genetic variation into a population of fungi. In fungi, sexual reproduction often occurs in response to adverse environmental conditions. During sexual reproduction, two mating types are produced. When both mating types are present in the same mycelium, it is called homothalic, or self-fertile. Heterothallic mycelia require two different, but compatible, mycelia to reproduce sexually. Although there are many variations in fungal sexual reproduction, all include the following three stages, figure 24.8. First, during plasmogamy, literally, marriage or union of cytoplasm, two haploid cells fuse, leading to a dikaryotic stage where two haploid nuclei coexist in a single cell. During karyogamy, nuclear marriage, the haploid nuclei fuse to form a diploid zygote nucleus. Finally, meiosis takes place in the gametangia, singular, gametangium, organs, in which gametes of different mating types are generated. At this stage, spores are disseminated into the environment. Link to learning. Review the characteristics of fungi by visiting this interactive site. http://openstax.org/l/fungi_kingdom from Wisconsin Online.